of the origin. So I am correct. First DNA a protein which bind with the origin, especially on the 9 base pair region, and then DNA B protein and DNA C protein. These protein come, and first of all DNA C protein recognize where the DNA A protein has already bind, and then DNA B protein, that is a helicase enzyme that break the hydrogen bond. So both DNA strand will be separated, and then SSB protein that come and bind with the single strand of the DNA that prevent the recoiling of the DNA, and again RNA primer come, RNA primer which bind with the leading strand and lagging strand. On the leading strand, single RNA primer will bind. On the lagging strand, multiple RNA primer will bind. And in the next process, DNA polymerase third will come and it add the deoxyribonucleotide on the both strand. So in the leading strand, because only one primer is present, so deoxyribonucleotide will added in the continuous manner. So continuous strand will be formed. But in the lagging strand, because multiple RNA primer is present, so deoxyribonucleotide will be added. So DNA will be formed in the fragment. So these are fragments called okazaki fragments. And then DNA polymerase first one come and it will remove the RNA primer, and instead of RNA primer, it will add the deoxyribonucleotide. And at the last, ligase enzyme come and just seal the nick or by making the phosphodiester bond. And at the last, there is a termination. On the termination, there is a ter site is present. So DNA strand both move in the opposite direction, and it will bind with the ter site, and the replication will inhibit. So that was the story of the replication in the bacteria, especially I have described in the E. coli bacteria. Now we'll start the replication in the phage virus or phage lambda virus. So all the steps are similar, only the difference is the initiation process. So I will explain how the initiation occur in the phage lambda. So in the phage lambda, the origin is called origin lambda. As in the bacteria, the origin was called origin C. And in the phage, the origin is called origin lambda. So this is the origin lambda. Origin lambda is present in the phage virus, and it has two sites. One lambda O site and second site which is rich AT rich region is present on this region. So origin will always contain the AT rich region. Like in the bacteria, the 9 base pair region and 13 base pair region were present in the origin, and AT rich region is present on the 13 base pair. So that is why in the replication process, first of all, DNA strand separation occur from the 13 base pair. So the function of 13 base pair first initiate the replication. That means first both DNA will separate to each other from the 13 base pair region. In the same way, in the phage virus, separation occurs start from the AT rich region of the origin lambda, because AT has made the two hydrogen bond, so it will be easy to break. If the GC is present at that region, that will be difficult. So DNA replication always start from AT rich region. That means both DNA will separate to each other from the AT rich region. So in the same way here occur. That means the origin of the replication of both DNA separation occurs from the AT rich region. So in the initiation process, first protein that is come into the lambda phage lambda virus is the lambda O protein. So this is the lambda O protein is coming here, and lambda O protein come and bind with the DNA. So this is lambda O protein come, and in the second region, if you remember that I can also compare here. In the bacterial replication, first protein कौन सी आई थी यहाँ पे, ये बताएगा. ई कोलाई में लैम्डा ओ की जगह कौन सी प्रोटीन आई थी सबसे पहले?
DNA A protein.
DNA A protein आई थी. Very good. So in the phage virus, lambda A protein की जगह lambda O protein आएगी. Then second step in the bacteria, the protein जो आई थी वो आई थी आपकी DNA B protein and DNA C protein. And in the virus, this is a two protein also come, DNA B protein, and in place of DNA C protein, that is protein called lambda P protein. So lambda P protein plus DNA B protein, both protein will come and bind, and the same function as in the bacteria, like the C protein will recognize the AT-rich region. So in the same way, lambda P protein which recognize AT-rich region, while DNA B protein, that is a helicase, so it will bind. So first these protein will combine and then bind with the DNA. So lambda B protein is works in the same way. It breaks the hydrogen bond on lambda P protein. It will recognize the origin and especially where the lambda O protein has already bind. So this is the second step. Now this is the third step. So in the third step, three proteins will come, and these proteins are called heat shock protein. So heat shock protein, that are the protein which, as the name suggested, heat shock. That means it prevents the DNA from the heating or from the damage. So that is called heat shock protein. So although heat shock protein has multiple functions, so here the function of the heat shock protein is just helping in the replication process. So it will bind and it will help to bind of the DNA B protein and DNA C protein properly on the DNA. So now here third protein DNA J, DNA K and DNA GRPE protein, these heat shock protein will bind with the DNA and this complex will form. And in the next step, single stranded binding protein, same, it is same here as in the E. coli, that is bind with the single strand of the DNA and prevent the recoiling of the DNA. It is the same function. So when the SSB protein bind, it is ATP dependent manner. That means when the SSB protein will bind, then ATP will be utilized. That means ATP is required for this step. So ATP breakdown with the ATP, so energy will release. So with the help of this energy, SSB protein will bind. And in the same step, these heat shock protein will be removed. So DNA J, DNA K and GRP protein will be removed. So finally, what is remaining here? This is the DNA A protein, equivalent is lambda O, and the DNA SSB protein and DNA B protein, that is a helicase. So finally, this structure is last in the initiation of the DNA. And in the elongation process, same things, same process occur. That means first RNA primer will add, it will bind with the both strand of the DNA, leading strand and lagging strand in the same way. Then DNA polymerase thread will come, it will add the deoxyribonucleotide, then DNA polymerase will come. It help for the proofreading and it will remove the RNA primer and add the deoxyribonucleotide, and then DNA ligase that will form the phosphodiester bond and seal the nick and the termination. The same process occur. So I hope आप लोगों को समझ में आ गया होगा. सबको समझ में आ गया ये? So Anmol Adhikari, are you present?
Yes, sir.
Anmol, I always that mail. Okay, Anmol, can you explain this process?
Yes, sir.
Okay, explain.
So in phase lambda, replication start with initiation process. And this in lambda, in phase lambda, origin is known as origin lambda.
Okay.
So or it also have two sides, lambda O side or AT-rich side. So and separation started from AT-rich side. DNA replication start from AT-rich side. So and the first protein in this phase lambda is lambda O protein, which come and binds with the DNA. So and then next protein come is DNA B and lambda P. So lambda P is the protein recognize the AT-rich side, and DNA B breaks the hydrogen bond. It is also known as helicase. So then three protein will come, which are heat shock proteins, DNA J, DNA K and GRPE. These are used to prevent DNA from heating or damage. So then SSB bind come, single stranded binding protein, and prevent the recoiling. and they remove the 3 heat shock protein, and the last remaining protein is DNA B, lambda O and SSB.
Very good अनमोल, बहुत अच्छा एक्सप्लेन किया आपने.
Thank you.
So I think अनमोल को समझ में आ गया अच्छे से, तो I think सबको समझ में आ गया होगा. आ गया होगा ना सबको समझ में?
Yes, sir.
अच्छा, एक ही आवाज आई कि चलो कोई बात नहीं.
Yes, sir.
अच्छा. Okay, next. Next is the replication in the origin P1 of the phase P1. So this is the another virus that is called phase P1 virus. So in the same way, process occur, or in this step, first we also see the initiation process. So in the initiation process, here there is 2 type of protein first come. That is a protein is called RAP-A dimer and DNA J dimer. So DNA J in the form of dimer. So that is why it is written in the two DNA J protein that form the dimer, and 2 RAP-A protein will combine and form dimer. So total is 4 protein. So RAP-J dimer and DNA J dimer both will combine and form the tetramer. And this tetramer first come and first make the complex. And in the next step, what happen? Same process, DNA K protein, GRP protein, and ATP. ATP is required for the addition of the DNA K and GRP. So these three component will come and bind with this RAP DNA J complex. And when this complex will bind, and then it will remove this DNA J, DNA K, GRP, ADP. That means ATP will convert it into the ATP. So basically, this function of the DNA K GRPE protein is here just remove the DNA J. So DNA J will remove, and other will separate the RAP monomer. Now I don't know why this process will happen, because maybe possible that RAP-A dimer, it can also come and directly bind with the DNA. But here the process, this is known process, so that's why I'm telling about the first RAP-A and DNA J dimer form complex, then DNA K GRPA protein will combine, and this protein will remove, and remaining is a RAP-A monomer. So maybe purpose of the conversion of the RAP-A dimer into the RAP-A monomer, then RAP-A monomer is separated, then RAP-A monomer come and bind with the DNA. So process start here when RAP-A protein will bind with the RAP-A site. That is a specific site is present on the phase P1, that is RAP-A site. So RAP-A monomer and bind with the DNA A site, and opening of the DNA from the DNA AT rich region. So in the next step is the same as in the E. coli bacteria, like DNA A protein. So here DNA A protein is coming, then again DNA B protein, that is a helicase, DNA C protein, SSB protein, single strand binding protein, then this is topoisomerase, then DNA G primase. DNA G primase actually that enzyme that form the RNA primer. So RNA primase enzyme, it is also called RNA primase enzyme or DNA G primase. So RNA primer, then DNA polymerase third, and that is some different like He protein, maybe histone element like protein, or this is the nucleotide RNTP, DNTPs, these all are the nucleotide. That means RNA polymerase will add these or nucleotide. That means all the process is same as the we have discussed in the E. coli bacteria. So from the DNA A protein, from the addition of the DNA A protein to the last, all this process is same. That means whole process that occurs in the E. coli bacteria, that will be happen from this step. And now this is the additional step when starting occur when RAP-A monomer bind with the RAP-A site. Then again this DNA A protein plus then DNA B plus C protein, then SSB protein, then DNA polymerase third, then first, all this process will be same. So you have to learn only this complex, that RAP before the addition of the RAP-A monomer. That means first RAP-A dimer and DNA J dimer will come form complex, then DNA K, GRP and ATP combined, and then wrap A monomers form, and this will bind with the wrap A site. So I think you can understand. So अब कौन तैयार है बताने के लिए? Any volunteers who is confident? कोई भी कॉन्फिडेंट नहीं है? यस,
Can I try?
या फर्स्ट आप नाम बताइए अपना.
Sir, Gunjan.
गुंजन, ओके गुंजन, ट्राई.
Sir, replication in origin P1 or fast P1. Sir, actually it is just similar to E. coli replication. तो इसमें क्या होता है कि REP A dimer और DNA J dimer दोनों आते हैं और वो combine हो जाते हैं. They form REP A and DNA J complex. Then उसके बाद DNA K and GRP E and ATP. DNA जो K होता है, मतलब DNA K प्रोटीन जो होती है वो हीट शॉक प्रोटीन होती है. सर वो उसके बाद जब वो कंबाइन हो जाती है, मतलब DNA K प्रोटीन और DNA K प्रोटीन जब वो कंबाइन हो जाती है, उसके बीच में ATP जो होता है वो ADP में चेंज हो जाता है. मतलब वहाँ पे यूटिलाइजेशन होता है एनर्जी का. उसके बाद REP A जो मोनोमर्स होते हैं वो ब्रेक हो जाते हैं. ब्रेक होने के बाद जो REP A मोनोमर जो होता है बाइंड हो जाता है REP A साइट्स पर. जब वो REP A साइट्स पे बाइंड हो जाता है, उसके बाद मतलब REP A साइट्स पे बाइंड हो जाता है. उसके बाद दैट अलाउज द ओपनिंग ऑफ DNA साइट्स A+T रिच साइट्स को ओपनिंग करता है. मतलब दैट अलाउ द ओपनिंग ऑफ द DNA फ्रॉम दिस साइट्स आल्सो. उसके बाद सर उसमें DNA A जो सर REP
रैप ए साइट बाइंड करने के बाद फिर आवाज नहीं आई मुझे.
सर एक्चुअली वो बीच में नेटवर्क प्रॉब्लम कर रहा था ना, इसीलिए.
अच्छा, हाँ, फिर रैप
हाँ,
ए प्रोटीन बाइंड करके उसके बाद फिर क्या हुआ?
DNA जो A प्रोटीन होता है, हाँ सर, DNA जो A प्रोटीन होता है वो REP A साइड से मतलब बाइंड होता है. जैसे E. coli में DNA A प्रोटीन बाइंड करता था, मतलब 9 बेस रीजन पर. ऐसे इसमें क्या होगा कि DNA जो A प्रोटीन होगा वो REP A साइट्स पर बाइंड करेगा. जब वो REP A साइट्स पर बाइंड करता है तो अलाउ करता है DNA A प्रोटीन को, मतलब ओपनिंग से, मतलब दैट अलाउ द A+T रिच रीजन फॉर रेप्लिकेशन. उसके बाद सर DNA B हेलिकेस, DNA C, ये सब मतलब इसके बाद तो मेन रेप्लिकेशन की जो इनिशिएशन जो प्रोसेस होती है फिर स्टार्ट होती है.
हाँ, आगे बता दो कैसे कैसे हुआ था उसमें.
सर उसमें तो इक्वलाई का लिया था ना सर, तो उसमें मतलब DNA C प्रोटीन होती थी. DNA C प्रोटीन जो होती थी वो मतलब दैट रिकॉग्नाइज द बाइंडिंग ऑफ द प्रोटीन. एंड जो DNA B प्रोटीन जो होती थी वो मतलब दैट अलाउ द ओपनिंग ऑफ हाइड्रोजन बॉन्ड एंड अलाउ द ब्रेकिंग ऑफ हाइड्रोजन बॉन्ड्स. तो उसमें ये प्रोसेस था सर. इसमें फिर वही प्रोटीन आए थे DNA B और DNA C. उसके बाद SSB प्रोटीन आती थी सर. SSB प्रोटीन मतलब सिंगल स्टैंडर्ड बाइंडिंग प्रोटीन. और सर वो दैट प्रिवेंट्स द रिकॉइलिंग. सर देन उसके बाद टोपोआइसोमरेज सेकंड दैट रिड्यूस द टेंशन. जैसे कहीं टेंशन हो जाती है उसमें गांठें पड़ जाती है, दैट प्रिवेंट टेंशन. फिर उसके बाद ये होता है. उसके बाद मतलब फिर इलोंगेशन की प्रोसेस होती थी, वही DNA स्ट्रैंड टेम्पलेट स्ट्रैंड को लेते थे.
ठीक है, वेरी गुड. ठीक है, you have done well tried. You and both Anmol both have very well done tried. Okay. So next same process as we have already explained in the E. coli bacteria, elongation, terminations, and now I can explain about the termination. It was remaining in the E. coli, so I'm just trying to explain you. There is a specific ter site present in the DNA where the termination occurs. So ter site is there is a ter protein like there is a two type of the two reason. First, TER-E, TER-D, and TER-A. These three proteins goes into the clockwise direction, and this is another is TER-C, TER-B, and that is a TUS protein. So this is bind here in the DNA in the anti-clockwise direction. So when the DNA coming in the clockwise direction and anti-clockwise direction, so when it reach to this TER site, so automatic this TER site will bind with the both DNA strand. So when it bind the DNA strand, so it will not further allow the replication process. So here the replication process will be stopped. So that means we can say that this TER site is responsible for the inhibiting the replication. Although there is not a well-known, well-recognized theory, but in the present scenario it is believed. The real mechanism is not known, but the scientists believe that these TER sites are responsible for the ending of the replication, or that is also called termination. So this is the just comparison in this slide. I am just telling you comparison of the different type of the organism. Although you have not to learn this, but for just for the knowledge, or if you want to learn, then it will be okay. Or if you don't want to learn, then it is also okay, no problem. So this is first helicase. So in the E. coli, the helicase enzyme is a DNA B protein, while in the archaea, that you know about the archaea, primitive environment, primitive bacteria or primitive organism, that is archaea. In the equivalent of the DNA B protein, this is protein called DNA A2 or MCM2 like protein. Or in the HeLa virus or SV40 virus, simian virus, in this virus, helicase protein is a T antigen. That means instead of the DNA B protein, T antigen is present here. And in the same way, in the bacteria E. coli DNA C protein, but in the archaea it is not known, but in the HeLa, that is a T antigen is same function. And the phage T4 virus, in case of DNA B protein, it is a G47 protein, and in place of DNA C protein, this is G59 protein is present. And now next one is a single stranded binding protein. So SSB protein, so in the archaea, in place of SSB, that is a RPA like protein, while RPA K-like protein, while in the HeLa virus RPA protein, and in the phage virus G32 protein. Next is a priming protein. Priming means primer formation. DNA G protein that form the RNA primer. So in E. coli DNA G protein or RNA primase enzyme form the primer, while in the archaea polymerase alpha like this form the primer, and the HeLa polymerase alpha or oblique primase, while in the phage G61P protein. Sliding clamp. Sliding clamp, I have discovered this. This is present in the DNA polymerase third protein, and the function of the DNA sliding clamp, just bind the polymerase enzyme on the DNA, so it will prevent the falling of the DNA polymerase third. So that means sliding clamp is a ring-like structure that bind with the DNA polymerase third enzyme when it will bind with the DNA and move on the DNA. So it will simply DNA polymerase enzyme को DNA पे लेके जाती है और उसे गिरने से रोकती है. And in place of that is a beta dimer, I have put that sliding clamp or beta dimer. Or in the archaea, it is a PCNA like polymerase chain nucleophilic antigen, and in the same PCNA in the HeLa virus, or in the eukaryotes, we will study about the eukaryotic replication. So we will also find that in the human PCNA is present. In the phase T4 G45P is present. And next is a matchmaker box, that is a gamma complex. This gamma complex is also called matchmaker box. It is also component of DNA polymerase third. I have explained in the previous class, DNA polymerase third is made up of core enzyme, sliding clamp, and gamma complex. These three structures represent the DNA polymerase third. So in the RFC-like protein, RFC and G44 are 62-like protein. Now, equivalent to the DNA polymerase third, in the archaea family B DNA, that is a protein which is equivalent to the DNA polymerase third. And in the HeLa virus, polymerase delta, that is equivalent to the DNA polymerase third, and in the phage G43 protein. Now another enzyme tau T2, that is present in eukaryotes normally, but say E. coli tau dimer. I have not explained about this tau dimer. This is also one of the component of the DNA polymerase third. So DNA polymerase third is a beta dimer, fork enzyme, gamma complex, and sometimes this tau protein also present, because in the every time tau dimer is not present, so I have not explained, but in some conditions, some bacteria, this tau protein is also present. But in E. coli, in the archaea and HeLa virus, there is not known any protein which is equivalent to the tau dimer. So that is why I have told the main function is of tau dimer is not known. And next one is the DNA polymerase first in the E. coli, and equivalent of the DNA polymerase 1, that is a FEN1, FAN1 or MF factor, maybe maturation factor it may be. So MF1 protein or G43 protein. And the ligator last is that one seal the nick ligase enzyme, and the same enzyme is present in the archaea, that is also called ligase, but in the HeLa that is called ligase T, and it is T4 ligase. Now the summary of this table, that in the different organisms, the same enzymes may be present in the different form. That means some protein will work of the equivalent of this protein. So that is why first I have explained the replication, the E. coli bacteria. So you have remember carefully of this all the protein, then you can compare with the another different protein. If you don't remember of this protein, then how can you compare? So that is why I explain many times that, so that you can learn properly replication in the E. coli bacteria. So you have not to learn all these things. You can just learn some rememberable things, like this is some important protein, like that is this one. This is a beta dimer, and in place of beta dimer, PCNA like protein, that is important. You can learn this PCNA, and a simple protein learn कर सकते हो, जैसे ligase है, ligase के ligase है, यहाँ पे ligase T है, T4 ligase है, that will be easy for learning. So you can learn this. That means ligase T होगा and T4 ligase. तो ये इसको learn कर लीजिए आप. और DNA polymerase थर्ड जो है, इसकी इक्विवेलेंट पॉलीमरेज डेल्टा होता है, दैट इज इम्पोर्टेंट है. DNA polymerase डेल्टा एंड DNA polymerase थर्ड. Beta dimer and beta dimer equivalent PCNA and PCNA, that is also important. And all these are not too important. T antigen you can learn that DNA B and DNA C protein equivalent यहाँ पे DNA 2 like protein होती है, यहाँ पे T antigen होता है, that's it. तो 2-3 प्रोटीन यू कैन लर्न कर सकते हो. टी एंटीजन कर लो, पीसीएनए कर लो, और यहाँ पे लाइगेस कर लो, और डीएनए पॉलीमरेज डेल्टा कर लो. बस इतना याद कर लो, और बहुत ज़्यादा लर्न करने की ज़रूरत नहीं है आपको. ठीक है, तो दैट इज क्लियर है आप लोगों को सबको. So this was the replication in the E. coli bacteria, phage virus. So now I will start replication in the eukaryotic species, including the human. Although replication is in the eukaryotes is too much complex, and there is not too much known about the eukaryotic replication. So some specific structure, specific things is only known about the replication. So in the next class we will discuss about the replication in the eukaryotes. So before I am starting the replication in the eukaryote, first you can just remind all these replication in the bacteria, replication in the virus. So I will send today the slides. You can learn this replication in prokaryotes completely, and you have not to learn too much in the eukaryotes. Just in the bacteria replication occurs from the origin. The origin will also present in the eukaryotes also, but this origin have a specific name like DNA bubble. So what is the DNA bubble? When the origin, that means spherical like structure, that will open, so it form the replication bubble like structure. That is, you can study in the eukaryotes, or you can also study this in the bacteria. But now another difference is like single origin is present in the E. coli or prokaryotes, but human DNA or eukaryotic DNA is very complex. So multiple number of origin is present in the bacteria. That means there is suppose 10 meter DNA. So in the 10 meter DNA, many origin are present and they will form the double stranded DNA. That means अलग अलग जगह से DNA का formation हो रहा होगा. ठीक है, that means multiple replications occur in the same time.